Hello everyone, the title of my final project is the neuroethology of ant foraging. Let's start it. And my presentation will divide into four parts. The first one is introduction. It will tell why I choose this behavior. And the second one is analyze. And this will use the psychological complexity and the Timberger form that was taught in class to analyze this behavior. And the third one is researches. This will describe some of the current researches, and the experiment is an experiment I designed to further investigate the behavior. Let's get into the first part, introduction. Why I choose this topic? The story begins in a corner of the Crossroad cafeteria one afternoon while preparing for the final project. I saw a group of ants foraging around some food crumbs at the table, and I follow the line they formed and find an anthill in the corner. So how ant forage and how they organized? This interesting question coming to my mind, and so I go to search for it. And let's start from some introduction. The foreign behavior of ant is complex and organized social behavior, which mainly involved searching for food acquisition, information exchange, and transporting food back to the nest. And the second part is analyze. In this part, I'm going to analyze behavior from perspective of psychological complexity and Timberger form. In the class, we layered the psychological complexity for the ant foreign behavior. It includes, for the ant foreign behavior, it includes three of the layers mentioned in the class. Innate behavior, learner behavior, cognition. Well, it's unlikely that the foreign behavior of ant involved such advanced psychological complexities as consciousness or theory of mind. Their behavior does suggest a degree of learning and cognitive ability. For they are typical social insects and always be considered as superorganisms. And in addition to the psychological complexity, Tinberger's four questions are often used to analyze different animal behavior during our class. And today I want to focus on the mechanism. Is the question, how does the foreign behavior work? So the answer is the question described how ants use their autonomy, physiology, and regular mechanism to perceive that their environment, process information, and perform the foraging task. So we will focus on these things. And let's get into the third one is the researches. What is known about it by researchers? And in the introduction part and the previous analysis, we can know that these most important part of the ant foraging behavior are navigation, recruitment, and regulation. Navigation requires the navigator to find food and record the return journal. And the recruitment requires navigation to communicate with the other ants after finding the food and achieve the purpose of transfer and process the information. And the regulation requires the communication among the colony to determine the hunger state of whole colonies, determine whether individuals need to go out of the nest to forage for food, to carry food, or to go to which food point to collect food in order to improve the efficiency of carrying. The study of this process made it more familiar with behavior of ant foraging, this behavior that's seen everywhere. First, we need to understand the ant sensory system, which is the basis of all physiological activity. And every part of the foreign behavior, include navigation, recruitment, and regulation, requires the involvement of the olfactory sensory system. Chemical communication is the original and perhaps the most efficient way of transferring information between the living unit and the research it pick in the superorganism, just like the ants. The ancestry of chemical signal provides the basis for the complex socialization and the long-distance foraging. In the ant nervous system, olfactory information is processed by two subsystems. Signals from the basic sensory are processed in the T6 glomerular cluster, and the other olfactory cues are processed in the mean AL area. And let's go to the navigation part. Navigation can be divided into two parts. They are food searching and returning to the nest after finding food. In the former process, foraging ants usually first travel through the side wing to get more smell of food, and then search for food by looking for smell. After finding food, foraging ants navigate by combining self-centered path integration and knowledge of landmark, orient themselves by observing polarized light pattern in the sky, spectral patterns, and wind direction, and rely on the internal counting of walking distance, just like the step integrator, to assess how far they are traveled and get a better determination of the closed distance traveled from the return for the return trip. And let's talk about the recruitment. There are four main ant recruitment model. The first one is mass recruitment, in which foraging ants or scout broadcast guidance information to their netmates in the form of a scent trail by having some pheromones on your way back to the nest, on the way back to the nest. And the second one is tandem running, where a successful scout lead a single recruit, just a single recruit, to the resource finding earlier. And the third one is group recruit. Scouting ant may lead a group of several individuals to the goal. And the fourth one is leader scouting system, in which a scouting communicates a distant location to foreigner. In this system, message about distant goals come from the scouting ant without relying on any other cues, such as order trails or direct guidance. And let's go to the last one, the forage regulation. In this part, I want to discuss the personal behavior and the superorganism, just like the group behavior. And the personal behavior may be related to an individual physiology with linear individual making extra foraging trip in response to an increased demand for foraging. And a study on ant colonies and individuals have found that the regulation of foraging behavior is a part of superorganisms' uptake of nutrition, in addition to the individual's behavior of individual's ant in the perspective of superorganism. So group personality of the particular configuration of behavior expressed by the group is likely to emerge from differential aggregation of individual personalities comprise the colony. In the case of swarming, ants increase the collective food intake by recognizing their nestmate through the destiny of recognition and the concentration of the chemicals that determine whether or not to participate in foraging and the prioritization of different destinations through this way. Okay, let's get into the fourth part. I want to express experiment of myself I want to do to further the research of the behavior. So the current research, the current studies treat the colonies as superorganism, in which the position of each individual's ant is more like a component of the organism, and the study is more the study of more detailed neural mechanism and circuit is not really clear. For example, in the homing stage, which brain region received the signal of barrier latitude, and in which nuclear of brain region of the integration and final decision making take place, and whether there is a circuit similar to the integration and the reuse of the variable information by bat in echolocation, just like we learned in the class. And now them is clear right now. So I hope to design maybe design an experiment to investigate the relevant circuit mechanism. And this is my experiment, and the purpose of my experiment is to verify the role of different brain regions in homing behavior, and especially the neural mechanism involved in receiving and in integrating navigation information. And the experiment setup we need is a spherical treadmill, including a stereotaxic device designed for ants, as shown in the figure, just this one. And it referred to a similar research setup for mice. The ant can be given a specific verbal signal via video on the screen and olfactory signal via deliver service that simulate the smell in the environment, and other signals such as a polarized light to simulate the ant returning to the nest after finding food. And the experiment procedure and consequence and conclusion is that I want to do this loss of function experiment by gene editing technology, just like CRISPR-Cas9, or use some microsurgery, causing functional damage to a specific brain region or nucleus of ant, comparing with the normal ants during the simulate homing process, observing neural activities and behavior abnormalities exhibit in specific brain region, and finally understanding the function of specific brain regions or nucleus in foreign behavior. By studying multiple brain regions or nucleus, the finer neural circuit can be summarized, maybe summarized throughout the forage behavior. Oh, that's all. And these are my reference for my presentation. And thank you. Thank you, everyone.